Hello, Johan. Can you hear me?
Hello?
Okay. I think we can start now. Is that all right for you?
Yes.
Okay. So, good afternoon, everyone. Welcome to today's talk on characterization of enzyme in amino biosynthesis in Catharanthus roseus. My name is Purin Chandra Purnama, and I'll be your moderator for the next 1 hour. So before we start, let me quickly to go over today's agenda. So we are going to have like 4 sessions. The first session will be introduction and then presentation. The third session will be opinion and question and answer. And the last session will be closing. Uh, for the first session, let me introduce our guest speaker for today. So Professor Johan Memelink. So he's the leader of plant cell physiology lab, which mainly work with finding genes for enzyme and transcription factor, regulation of transcription factor, and also expression of enzyme at gene and protein level. And he's here today to share his expertise with us. So without further ado, I'll hand it over to Johan for the presentation. Please, Johan, you can start your presentation.
Okay. Good morning, good afternoon, everybody. It's morning here. Okay, so Purin asked me to tell you something about our work on characterization of enzymes in the alkaloid pathway in the plant Catharanthus roseus, which we finished about 10 years ago. And just to introduce my own interests and the topic in general. So I'm a professor in plant cell physiology at Leiden University. And my research interests are, as Purin already introduced to you, is finding genes for enzymes and transcription factors that are involved in secondary metabolism, and looking at the expression of enzymes at the gene and at the protein level. And I was actually most interested in how genes are regulated by transcription factors. So these alkaloids belong to the large class of natural products from plants. So these are called natural products or secondary metabolites or specialized metabolites. And in general, they are organic compounds of molecular weights around 1000 Dalton. And they have important roles in plants. So they have important ecological roles in defense and communication with other organisms. And for the alkaloids in my plant of study, the Catharanthus, several of these alkaloids are also valuable as pharmaceuticals for humans. Okay, so these monoterpenoid indole alkaloids are found in a rather large group of plant species. An important species is this Catharanthus roseus. The former name is Vinca rosea, so it has been renamed a couple of decades ago. Let me see. Somebody has the microphone open? Okay. The English name is the Madagascar periwinkle, and the name comes from their biosynthetic origin. So they are alkaloids with a terpenoid part, which is here indicated in blue, and an indole part, which is indicated in yellow. So they have two biosynthetic origins. And the compounds that are shown here are two monomeric, as they are called, MIA compounds, which are used as medicines against hypertension.
Wait, Johan?
Yes?
Wait, something wrong happened with the presentation slide because we are now still watching the first slide.
Oh, yeah?
Yeah.
Okay. Um, so you're still seeing the first slide?
Yeah, but now it's already. I think this one, the Mia metabolite, is that the one that you are explaining, right?
You are seeing now a blue slide with compounds?
Yes, yes, blue slide with compound. Yeah,
Okay.
from the Mia. Yes.
Okay. And now you are seeing something with medicines?
Yes, yes,
Okay.
correct.
Okay, well, let's hope it works.
Okay.
Okay, so I was talking about this slide that you're now seeing. So you see that the compounds have a yellow part, which is the indole part, and a blue part, which is the terpenoid part. And to be more precise, it's a monoterpenoid part, as it is called. And then the top ones are the monomeric medicines from Catharanthus. And then the plant also makes dimeric alkaloids. So there's two of these monomeric alkaloids are fused to each other. And the two compounds that are shown here are important anti-cancer medicines, vinbestin and vincristine. Okay. Well, this is just a slide to show that.
Recording in progress.
Okay. I'm being recorded also, Poorin.
Yes, yes, that's
Okay.
correct.
Okay. Okay. So this is to show that there are several trade marks for these compounds. So at the bottom are the two natural compounds, vincristine and vinblastine, and they have various trade names as medicines against cancer. And then in the top part are more recent derivatives of these natural compounds, so they are modified compounds, and the modified parts are indicated in the red circles. And they are also sold as medicines under different trade names. So this is an important product from this medicinal plant. Okay, and so the work that I will tell you about was done as part as a collaboration in a larger project, the so-called Smart Cell project, which was an European Union project financed by EU. And it has had several academic partners from different countries in the EU. So in total, 14 academic partners. And we had also some commercial companies that were involved in the project. And the idea of the project was to elucidate the seco-iridoid pathway. I will explain to you what this is in Catharanthus roseus and try to engineer it in this plant and in other plant species to Nicotiana species. Okay, so at the start of the Smart Cell project in 2008, only a limited number of genes encoding enzymes in this pathway were known, and they are indicated here in the blue blocks. So these are all the enzymes, and which was at the time, say, really in the pathway. So this is actually a precursor pathway. In the top, the MET pathway. And at the left, you also see the shikimate pathway, which is also a precursor pathway. So in the pathway itself, about 15 enzymes were known at the time. What you see in the right part is a pathway going from geraniol to secologonin. And this pathway is called the seco-iridoid pathway. And a few genes were known in 2008, but a large number of genes were still unknown. And the idea of the Smart Cell project was to solve this part of the pathway. Okay, so the pathway goes from geraniol to secologonin, and geraniol is the monoterpenoid that delivers the terpenoid part of the indole, the monoterpenoid indole alkaloids. And I've indicated here the known genes. So there was one gene known that catalyzes the first step, the hydroxylation of geraniol. And you see here two genes indicated actually, and that is because this enzyme. Okay. So this G8O enzyme is a so-called cytochrome P450 oxidase. And this kind of enzyme has a coenzyme, and this is the coenzyme. So this is in fact one enzyme complex. And here at the bottom you see two enzymes for the last two steps. And everything in between was unknown in 2008. Okay. So what was the strategy? So what we did is we started to deduce the most likely pathway to go from geraniol to secologonin at the chemical level, and then kind of suppose that there are certain enzymes that catalyze these steps, then identify the genes for these enzymes from all kinds of transcriptomics data, and then isolate these enzymes and express them in different organisms, and then purify, in some cases, the enzyme, and then do assays with the enzyme to see whether they indeed catalyze the enzymatic step. Okay, so what we came up with is this pathway. This was just chemistry on paper, looking how in different steps geraniol could be converted to secologonin, and also known that these compounds that are indicated here occur in Catharanthus roseus, so that they could be possible intermediate compounds in this conversion. So what I've indicated here is a little bit more than in the previous slide. So besides the known enzyme, I also indicated a number of enzymes that were described. And what I mean by enzyme described is that the enzyme activity was reported in a plant extract. So certain researchers made plant extracts from Catharanthus, and then they did an enzymatic assay and they could find this enzyme activity. So that shows that the enzyme activity exists, but you still do not have the gene. So still, this was still a target for the Smart Cell project to isolate the genes for these enzymes that were described. And then we hypothesized that there are 3 unknown enzymes involved in the conversion of this iridodiol to 7-deoxyloganetic acid. Okay, so this is the same kind of pathway, but then at a more chemical level. So yeah, this is the way we were looking at this pathway. So just to see, okay, what are the different steps? And by looking at what chemically happens, so for example, here in the first step of where there's a described enzymes, the HGO enzyme, there's a dehydrogenation, so removal of water. So that also gives you an idea what kind of enzyme you are looking for. So these enzymes are conserved enzymes, conserved proteins, which in principle you can recognize in a DNA database if they occur. And then the next step indicated by MTC is a cyclization reduction enzyme. And then the next step, the unknown step, is a hydroxylation. So that you know that you're looking for an oxidase enzyme. So this kind of looking at this kind of chemical conversions gives you an idea what type of enzyme you are looking for. Okay, this is the last part of the pathway, so it's the same story. So, for example, here at the DLGT step, there's a glucosylation happening. So you know that you're looking for an enzyme that belongs to the class of glucosylases. Okay, so
Iya, Pak.
here I indicate.
Keluar.
Okay. So here we are looking again at the same pathway where I indicated the known enzymes in black and the missing enzymes that we were looking for in the Smart Cell project indicated in red. So potentially 7 unknown genes, and they are all part of a consecutive part of the pathway. Well, so looking at the chemistry again, we kind of determined what type of enzyme we are looking for. So the first step in the upper left is cyclization, and so we are looking for a cyclase, and from the enzyme chemistry that was known, we also know that the enzyme uses NADPH, so that it is an NADPH-dependent reductase. And the next step to the right of that is a hydroxylation. So there's a hydroxyl group added at the top. And there's a question mark here for the enzymes, whether it's a P450. So P450 is an enzyme of the class of cytochrome P450 oxidases, which is a very common class of hydroxylation enzymes in plant secondary metabolism. So that's why we assumed that it might be a P450 enzyme. We went on like this to kind of find out what type of enzyme we were looking for each of the steps. This is just an example for one particular step where 8-oxogeraniol is converted into iridodiol. So you see that a cyclization is occurring, and this is the possible reaction mechanism. So this enzyme also uses NADPH to form this product. So iridodiol can exist in two forms. So what we see is that there's a ring formation, and the enzyme uses NADPH and reduces a double bond. So so we know that we are looking for an NADH-dependent reductase cyclase type of enzyme. Okay, so once we had figured out how many possible enzymes we were missing and what type of enzymes they were, we have looked in sequence databases for enzymes that belong to the classes that we have determined. And then the idea is that once you characterize the enzymes, you have candidates, and then you want to test the activity of the candidates. And this can be done in vitro or in heterologous systems or in other organisms to express these enzymes, or it can be done in planta via expression in plants. And then the final step is to test the enzyme activity. Okay, the way that we looked at or what helped us very much to find candidates is also a strategy that is called the co-expression analysis strategy. And this strategy depends on the assumption that the genes in the metabolic pathway, such as the seco-iridoid pathway, have the same type of expression pattern, so they have the same type of regulation. So what you need for this strategy is to have at least one gene in the pathway, so that you can look at the expression of this known gene, and then you can search for unknown genes that have the same type of expression pattern. And then the combination of the expression pattern and the fact that the unknown gene encodes the enzyme that you're looking for makes it a good candidate for an enzyme in this pathway. Okay. So we had two known genes in the pathway. So the first two steps in the iridoid pathway, the geranyl synthase and the geranyl 8 oxidase. And we had several types of plant material to look at the expression. So the strategy works best if you have as many different types of plant tissue where you can look at the expression, so that the correlation gets as good as you can get it. So we had induction by defense hormone, and we had different tissue types. So we could look at the tissue specific expression of the genes. So for these conditions, we had expression databases. And just to show you an example for one of the new enzymes candidates that we found, is here you see an expression pattern in different types of tissues. So the tissues are indicated at the top. So there's several cell suspension types, and there's several seedling types that were treated with jasmonic acid. And the level of expression is indicated by the color. So blue is high expression and yellow is low expression. And indicated are several known genes. So STR1 is a known gene that is not involved in the iridoid pathway, and then G80 and GES are two known genes in the iridoid pathway. And then we have here a candidate, ADH10, which has the same expression pattern as G80 and GES, and very different from the STR1 gene. So that is in principle a candidate, and it is also an alcohol dehydrogenase, so a dehydrogenase type of enzyme. And it turned out that this was the gene encoding the enzyme 8-HGO, so one of the enzymes that we were looking at. Okay, so by looking at the expression data and at the type of enzymes that the candidate genes were encoding, we found a number of candidates for the different steps. So sometimes quite a large number of candidates. So we had 11 candidates for oxidoreductases, of which several were tested. We did not test all of them actually. Not all of them had the most perfect co-expression pattern. And here, for example, we had 12 candidates for cytochrome P450 hydroxylases, which could be candidates for any of the 3 different steps here, because these are all hydroxylation, and therefore all these candidates can be candidates for any of these steps. So in principle, these candidates were all tested at the enzymatic level to see whether they were encoding the enzyme that we were looking for. In different ways. So the non-cytochrome P450 enzymes were produced in E. coli in a protein production system, and then purified by affinity chromatography and then tested. And then the cytochrome P450 enzymes are kind of complicated enzymes because they also need a coenzyme. So it's not possible to use the E. coli system. And there 2 different systems were used. So they were either produced in yeast or in a expression system in Nicotiana benthamiana, so in plants. And they were not purified, but assayed in crude extracts. So in principle, then the different enzyme preparations were tested with the hypothetical substrates. And then what helped us a little bit with identifying the whether there was a reaction occurring is that many of the steps produce or consume NADPH because they are hydroxylases or NADPH dependent enzymes. And NADPH has a high absorbance at 340 nanometers. So that means that any enzyme that produces or consumes NADPH, you can easily follow a reaction whether a reaction occurs by just measuring the absorbance at 340 nanometers. And then finally, if something was happening with the NADPH consumption, for example, then products were measured by HPLC or GCMS, and finally by NMR. Okay, so I've talked about the genes and then about how to produce the enzymes for testing. But an important step is also that you need the substrates and preferably also the products to be able to characterize whether the exact reaction that you are supposing is occurring. And there you run in another problem, and that is that many of the natural compounds from plants are not commercially available. So I've indicated here the different compounds in the pathway and which ones are commercially available. Those are the ones in yellow. So geraniol, the first compound, and the two last compounds, loganic acid and loganin, and actually sacologanin, are commercially available, but the others are not commercially available. Then we in Leiden had the four compounds that are indicated in green in the top, and these were previously synthesized before the start of the Smart Cell project by PhD students in my group. And then all the compounds in blue were synthesized by a company, and they were synthesized from another iridoid compound called alcubin. So it's actually not possible or very difficult to synthesize these compounds just from scratch or from geraniol, say, chemically, because these reactions are quite complicated. And not so easy to reproduce in a chemical lab. So that's why the company wanted to use alcubin, so a natural product, as the substrate to convert into these different compounds. So that's a quite common method to synthesize complex compounds. So we looked in literature. This was quite old literature, actually from the 60s, how you could make the different compounds from alcubin. So here at the left you see alcubin, and then via different relatively easy chemical conversions, you can make the 5 different compounds that we were looking for. And we found this chemistry in articles, as I said, from the 60s. And then we went to a chemical company in Holland called Chiralex, and we said, can you do this? Can you reproduce this if we supply alcubin? And they said, yes, we can do this. We can make 100 milligrams of each of these compounds if you give us 10 grams of alcubin. And alcubin is also not commercially available. So my PhD student then went on to actually isolate alcubin himself. And this photograph is of the plant Aucuba japonica. So it's a common, it's actually a Japanese shrub. It's a common garden plant in the Netherlands. And here you see the one that we used for harvesting the leaves. And this shrub is standing in front of one of the university buildings very close to our lab. So this Aucuba japonica has a very high percentage of alcubin in the leaves. So 2% of the wet weight of these leaves is alcubin. And so we went on a Saturday morning when there's nobody around. with a large shopping bag, we went to this shrub and we just harvested the leaves. So we harvested 10 kilos of leaves. And then we went to the lab, and there the leaves were extracted, and the alkubin was absorbed to charcoal, and then purified by silica gel column chromatography. And then we obtained, my PhD student obtained indeed exactly 10 grams of pure alkubin, and the purity was determined by NMR. So this is what we gave to this chemical company, Chiralix, and they used it to synthesize our 5 compounds. So you see that for doing this kind of experiments is not so easy, because you're not only looking for the genes, but to test the genes, you also need compounds that you cannot commercially buy, and that you actually have to make or have to order at a chemical company that can make them. Okay, so when we had all the possible substrates and products, we could do our enzymatic assays. So this is an example of one of the enzymatic steps. So we had a candidate for the conversion of 7-deoxyloganetic acid to 7-deoxylogenic acid. So it's a glucosyl transferase, so it attaches a sugar group here to the molecule. And this enzyme was found by this co-expression analysis method. And indeed, one of the candidates catalyzed this chemical conversion. So here we have the chemical analysis. So here in the top 2 runs of this HPLC are the substrates and the products, and the bottom run is the substrate with the purified enzyme. And you see that actually this enzymatic reaction is very efficient because the substrate 7-deoxylog. I think I said it the wrong way around. So the middle one is the substrate, the top one is the product, and the bottom one is the reaction. And you see the substrate is completely converted to the product. So this enzyme is very efficient in doing this reaction. Okay, here is another enzyme that we found. Other candidates, and this enzymatic activity was measured by our French partner in Strasbourg. And it turned out that this was an interesting enzyme because this was a cytochrome P450 enzyme, which could catalyze the conversion of iridodiol to 7-deoxyloganetic acid. And so that's the green steps. So we hypothesized that there were 3 enzymes involved, but actually it's only a single enzyme. So a single enzyme can do this quite complicated chemical conversion. So that saves a lot of enzyme characterization, because with this one enzyme we have this whole part of the pathway solved. Okay, here we have, so this iridiol oxidase then replaces these 3 different steps. So it's only actually a single enzymatic step. Okay, here we have hydroxylase enzyme. So that was also one of the candidates that was tested by the partner in France. It's also a P450 enzyme. And it does this chemical conversion. And then we have here the iridoid synthase, which makes this cyclic compound iridodiol from 8-oxogeraniol. Now, this enzyme, for this enzyme, we were scooped by the group of Sarah O'Connor. So they published in 2013, I think. We see 12 in 2012. They published this paper just about this single enzyme. It's also a special type of enzyme. So that's a pity, of course, that you're scooped by another group, but yeah, that happens. And then in the top, we characterized this enzyme in Leiden. So this was an oxidoreductase, which can catalyze the different conversions. So it introduces this aldehyde group. So there's two hydroxy groups which can be converted to two aldehyde groups, and this can occur either one first, and then the dealdehyde is the final product. So this enzyme was also coming out of the co-expression analysis and was analyzed in Leiden. And this is the chemical analysis of this enzyme by HPLC. So here we have the different compounds that were made by the chemical company Chiralex. And at the bottom run is the enzymatic reaction, where the substrate, 8-oxogeraniol, was incubated with the enzyme, and you see that all the different compounds are formed because the enzyme works in this type of grid. So it can convert the top substrate to any of the three products. And that is what you see here in this bottom HPLC run, that you can find all the different products. And all the substrates and products were characterized also by their mass spectrum, so that you can really determine that these are the compounds that we are looking at. Okay, so that means that after all this work, which was done, the enzymatic work was done in Leiden and in Strasbourg in France. We have all the missing enzymes in the pathway, so it turns out that we have 5 new enzymes, so genes for enzymes, and that we now have to complete pathway in principle on paper. And yeah, we were not completely satisfied with having this just like this on paper. So we also want to show that in planta, these enzymes can convert geraniol into secologonin. And for this, we used another type of assay, which is called an agro-infiltration assay in a heterologous host plant, Nicotiana benthamiana. And the way this works is that you introduce each of the genes that you want to test in Agrobacterium tumefaciens. So this is a soil bacterium which can transfer DNA to plant cells. And then you can mix the different Agrobacterium strains with the different genes and infiltrate them in plant leaves. So this is the way it looks. So the syringe contains the mixture of the Agrobacterium strains, and the syringe is pressed against the leaf, and then the Agrobacterium cultures are infiltrated into the leaf spaces. And in these leaf spaces, the Agrobacterium strains introduce the genes to the plant cells, and these genes are then expressed there in these plant cells. In this example, I just mixed 3 genes, but you can mix 10 or 20 genes in one mixture and then test the whole combination to see whether they can then convert a certain substrate to a product. So the advantage of this whole method is also that it's a relatively quick method, because after the infiltration, you can test the leaf extracts after about a week or so. Okay, so what we did is we infiltrated a combination of 12 genes. So these are all the genes indicated in this with the purple color. And with these 12 genes, we were able to show that geranyl diphosphate was converted into strictozidin, which is the first alkaloid in the pathway. So that shows that indeed these enzymes together can form a pathway in planta that can convert the substrate into the alkaloid products. So this was kind of the final proof that we wanted, that we indeed had the enzymes that we were looking for, and that we succeeded in solving this whole pathway. Okay, so all this work led to this paper with a lot of authors, of course, from the different groups that participated in this project. It's all the Smart Cell projects, which was published in Nature Communications in 2013. Well, actually 2014. It took a long time actually to get it accepted. You can see that it was submitted in 2013 and then published in April of 2014. Okay, and yeah, these are the collaborators in the Smart Cell project. You can also see that there's a large number of collaborators, and that they were active in different labs in Europe, in different countries, France, Belgium. And you can see that in the Leiden group, also Purnama contributed to the work, actually not to the work in this Nature paper, but in follow-up work that we published a year later. She also contributed to the work. Okay, this is my last slide. I thank you for the attention. I hope that you enjoyed the talk.
Okay, thank you very much for the very interesting presentation. I'm sure that we learned a lot from you today. To start, I have several questions for you, but because I'm very new in this research area, I have only a very basic question for you. So, as far as I know, in the past, the overexpression of a single gene in the pathway sometimes leads for some enzyme to an increased production of the direct product, but not necessarily to an increased alkaloid production. Yeah, the feeding precursor that increase the alkaloid production. So, is that problem already solved, or are we still struggling to solve this problem?
So what's exactly the problem that you want solved? It was not so clear to me.
Okay, so the overexpression of a gene, specific genes, we intend to increase the alcohol production, but in certain cases, it's actually the feeding precursor did increase the alcohol production. So the overexpression of genes sometimes didn't follow by the increase of the alcohol.
Yeah. Yeah, well, you see that the pathway is very complicated. So that means that overexpression of a single gene will not change too much in this pathway, I think. So what the group of Sarah O'Connor has done is they have expressed all the genes also in yeast and shown that they can produce the compounds in yeast. What's to overexpress transcription factors that regulate this pathway. And I think that's if you want to overexpress a gene to improve the expression of the pathway, probably the best is to overexpress the transcription factor that regulates the pathway. And that's also what we showed in this experiment, that by overexpressing one transcription factor, you can actually improve the production of the compounds. So that's maybe a better strategy. Although it must be said that not all the transcription factors that regulate this pathway are already known. So that's another problem maybe there.
Second question. What is your opinion about the works of hydroxylase isolation? Is it going to give any bright future, or there are so many competitors in this topic?
The isolation of what?
Hydroxylation enzyme?
Hydroxylation enzymes.
Yeah. Particularly from endophytic fungi, because several endophytic fungi can hydroxylate several secondary metabolites from their plants.
Yeah, well, these hydroxylation enzymes are in principle all very specific enzymes that catalyze one specific reaction. So I think that for each reaction that you are looking at, you need to isolate the exact enzyme that actually can do this. I mean,
Okay.
these are all very specific hydroxylation enzymes. So for example, 7-DLH does only this hydroxylation and not any other enzymatic reaction. So they are very specific enzymes.
Okay. What about salicylic acid hydroxylase enzyme? Salicylic acid hydroxylase enzyme.
Salicylic acid hydroxylation. What about it?
Because in my experiment, actually, we did a competing experiment, salicylic acid, into our endophytic fungi, and it seems that the endophytic fungi are able to hydroxylase the feeding compound.
Okay.
Do you think it has any bright future, or there are still too many competitors, and it's not something new in this area?
I don't know. Why do you think there are many competitors?
Yeah, because there are many established groups already did this experiment actually.
Okay. I do not have a particular opinion about this salicylic acid hydroxylation.
Okay, so better we try. Okay. Thank you very much. That's very helpful for me. Okay, now for students or maybe my colleagues, if are you guys going to ask a question? Please just raise your hand, or you can ask directly question to Johan. Please mention your name and also your affiliation. Okay, Mas Ibrahim, monggo, please.
Okay, thank you so much. Can you hear my voice?
Yes.
Okay. Just a quick question regarding the pathway itself. If I'm not mistaken, the cytochrome P450 enzyme is helping to clarify the missing steps in the cycloirradiate biosynthetic pathway. The quick question is, what the impact did discovery have on the pathway model? and can we use it as like a cheat code to find another missing pathway, another plan? Because as you say before, P450 are really, really universal enzyme, and almost all plant had it because it's really important enzyme. And yeah, that's the quick question. Thank you.
Yeah, so the P450 enzyme it's a group of enzymes. So it's one of the largest enzyme families in plants. So I do not know exactly how many P450 enzymes, for example, Catharanthus roseus has, but it's several hundred. So it can be maybe 500 or so, up to 700 different P450 enzymes. And they all are very specific. So each of these enzymes has a very specific substrate and catalyzes only a single reaction. And what distinguishes these P450 enzymes is that they are very conserved, so you can very easily characterize them in a DNA sequence database. You can immediately see, okay, this gene is encoding a P450 enzyme. But what you cannot predict is what the reaction is that the enzyme can catalyze. So that is something that you still have to determine in a biochemical way to see which specific enzyme reaction is catalyzed by a particular P450. So yeah, I think the work does not contribute something really to a model, because the model is already that most oxidations or hydroxylations in plant metabolism are catalyzed by P450 enzymes, and that's already known. That's already known for a long time, and so yeah, that certainly justifies to look at P450 enzymes whenever you're looking at an oxidation step in plant metabolism, but you still have to. I mean, since it's a very large family in any plant species of between 500 and 1,000 members, you still have to look for a specific enzyme if you're interested in a specific reaction.
Okay, Mas Ibrahim.
Okay, thank you so much. That's clear then. Okay, thank you.
Yeah, is that clear now? Okay. Thank you. Next. Any other question, maybe?
Yeah, what I maybe should add is that this agro-infiltration method has simplified also the analysis of the P450 enzymes. So if you have, for example, for a particular oxidation step, if you have 10 candidates, 10 P450 candidates, you can just introduce them in agrobacterium, mix them, and infiltrate them as a mixture, and then see whether they can catalyze a certain reaction. And then if you have a positive answer, you know that one of these 10 is the right candidate, and then you just can individually infiltrate each of the 10 to see which one is catalyzing the reaction. So this whole method actually simplifies the analysis of the candidates to a very large degree. So this method is really kind of a game-changing method in an analysis of these P450 enzymes.
Okay, Mas Ibrahim, is that clear enough?
It's clear now. Thank you so much for
Yeah.
the answer.
Okay. Who has more questions? We still have the expert here. Yeah. Any question? Okay. Yes, okay, that's also possible. I can also read the question from the chat box. Okay, Johan, maybe you can open your chat box, please, because we get question here. Yes, you can type.
Let me see. Can I see the chat box?
Yes, you're welcome. Yeah, okay, I'm waiting.
Yeah, is the question already there?
No, not yet. I'm going to
Oh,
tell you.
okay.
You're already there.
I
It's
see
not
this.
there. It's not there yet. My friend said okay. She wrote the question now. I think she's
Okay.
Okay,
now. Okay. Just wait for couple like 30 seconds. I'm sorry.
Yeah, it's
okay.
okay. It's okay.
So how long does it take for you to discover this five genes?
Say this again. How long?
How long does it take, yes, for the experiment in total to be able to publish
Well,
this new discovery?
the Smart Cell project was a 5-year project.
Oh, it's 5 year project.
And yeah, took we took quite a long time to do this. Just at the end of the project, we were able to reconstitute the whole pathway. So in that way, we were successful, and we were quite happy that actually the Smart Cell project was successful. Yeah, in the beginning, we had a slightly different approach, which was not working so well. So we lost some time, and then once we chose for the co-expression strategy and this co-infiltration, this ego infiltration strategy, this really speeded up things.
Okay. So the work is very challenging, right?
Yeah, so you can also see how many people were involved.
Yeah. Okay. So now we have the question. Can you
Yeah,
read?
I'm reading it.
Yeah.
Yeah, I have the impression that the answer is the talk that I just gave, or am I wrong? So
Yes.
so the co-expression analysis was very important for identifying candidates. And yeah, this co-infiltration of this ego infiltration method was very important for testing the candidates. But you see in different projects where people characterize novel pathway genes that they are very successful by using this co-expression strategy. So this is certainly a very powerful strategy for finding new genes in any pathway.
Okay, but Nino, is that clear enough? No, it's okay, no problem. Did you want to answer your question? You can type it. I believe. Okay, I think one more question. Is that okay, Ivan?
Yeah.
Okay. So, who has more questions today? Okay. We get the answer from Nina, so I think it's bit clear for her now. Okay. Another question from Faiza. He's one of our students. Can you read the
Yeah.
Question? Yes.
Yeah, how were these enzymes characterized? Yeah, so essentially we identified candidates by co-expression analysis, and then the genes were cloned in E. coli, expression plasmids, and then the recombinant proteins were isolated, purified, and then just analyzed in enzymatic assays with the supposed substrates. Yeah.
Okay. So, Mas Faizal, can you get the answer?
So, for example, this slide here shows an oxidoreductase, which was produced in E. coli. Yeah, so this HPLC run, the bottom one was just done with a purified, so a completely pure, more or less, protein, which was incubated with the substrate. Yeah.
Okay. Okay, I think it he has a better view to understand this process now. Okay, the next, Mas Alvaro. Yes,
Can
are
you hear
you?
my voice?
Yes, thank you.
Okay.
Can you hear me?
Can you hear my voice? It's a little bit noisy.
I'm sorry, but it's not clear anymore.
Okay. My my question is, where you assume that um P P450 is the gene that, yeah, it's common. Like, why you assume that? Like, what is the connection? Yes. Is my voice? Can you hear my voice?
Yes, I can hear,
Yes.
but I did not hear a question yet.
Oh yeah. Wait. I will type it.
Okay, you can type.
Oh, okay. It's also possible.
Yes, sorry. Okay.
Corinne, did you also distribute the Nature paper?
I'll only at several students. I'm going to distribute the papers to whole group
Okay.
because transcriptomics is a new thing for us. We start to study about metabolomic a little bit. Transcriptomic really nice.
Okay.
Yes, Mas Alvaro, can you type your question?
There's no new question in the chat box, is there?
No, not yet. I'm going to ask him. Wait for a second.
Okay.
Oh yes. So now we see this question.
Yeah. So it's about why you think that cytochrome P450 is involved. Yeah, because it's common. So essentially, most of the oxidation reactions in plant metabolism are catalyzed by members of the cytochrome P450 family. So that's why it's in general quite safe to assume that cytochrome P450 enzyme is involved if you're looking at an oxidation step. Yeah, so it's common.
Okay, because it's common. Okay, Mas Alvaro? Did you
Yeah,
get the message?
Again, it's also it's a large family, of 500 to 1,000 members in any plant species. So
Yeah. Okay. Is there any more question, or if you still have many questions about this topic, you can send via WhatsApp to me and I can send it to Johan. And if he doesn't mind, maybe he can give you the answer. Okay. Before we close this today talk, I would like all of you guys to on cam, because we are going to take a picture of us. Please on cam. Yes. Yeah.
Okay, I will take the picture.
Ya, bagaimana Mas? Sudah on cam semua?
Can all of you please turn on your camera? Kameranya dinyalakan.
Ya, tolong kameranya dinyalakan. Ya, Johan, could you please also present? You can close the presentation.
Professor, yes. Can you close the PowerPoint? Because I want to take a screenshot of everyone, and
Yeah.
the PowerPoint taking like a half of the screen. Yes.
let me see. Stop share.
Yes.
Yes.
Yeah. Oh, okay, nice. Oh, we can see each other now. Okay. Give the best smile. Sudah, Mas?
Say again.
Smile. Have you took the picture? Ya, Mas Ibrahim sudah tuh.
Yes. I already screenshot the first slide and second slide.
Okay.
Yeah.
So, okay, that's all. That's all.
I think that's all. That's all.
Okay. Okay. Thank you very much for all of your participation and engagement for today's talk. We hope you found this talk informative and useful. So we look forward to seeing you at our next event. And thank you very much once again for Johan, uh, for your very interesting talk, because this is something new for us. We learn a lot from this. Hope we can make more research collaboration in the future. And thank you very much for the students for the present and also my colleagues. Please have a great day and see you next time. Okay. Thank
Okay.
you.
Bye-bye.
Bye. Thank you very much. Oke, izin untuk menutup ya.
Recording stop.